Hello, I'm Dr. Aizaz from MedicoVisual.com, and today we will talk about vasculogenesis and angiogenesis. During the third week of development of the embryo, the blood vessels begin to form. So, as we have discussed previously in the lecture of gastrulation, that from primitive streak the cell ingress downward and they convert into mesenchymal cells. And as you know that this endoderm, it continues with the yolk sac endoderm, and it is further outside plastered with the extraembryonic splanchnopleuric mesoderm. Now what happens that these mesenchymal cells, they come here into this area, the primitive endodermal area, and they displace these primitive endodermal cells to the yolk sac endoderm. So here they are displacing it to the yolk sac endoderm, and now this primitive endoderm is almost completely replaced by those cells that came from the primitive streak, and then the more cells come into this space in between them, and as a result three germ layers are formed. Now what about this yolk sac endoderm? As we have discussed already that this yolk sac endoderm, through certain factors, it releases certain factor that causes the formation of blood island here. What I think of it is that this yolk sac endoderm, it is like a boss who orders the extraembryonic mesoderm that, hey extraembryonic mesoderm, I order you to start creating blood islands. And it follows this extraembryonic mesoderm, it follows the order of yolk sac endoderm, and it creates these blood islands. Now these blood islands, they result in formation of blood vessels as well as early or primitive blood cells. Now what are these blood islands? Of course they are not actual islands, so what are they? Basically, these blood islands, they are solid aggregates or clusters of special type of cells. So here you can see that this is a single blood island. It is a clump or cluster or aggregate, solid aggregate of these small tiny cells. And what are these cells? These cells are called hemangioblasts. This term hemangio, it consists of three words: hem, angio, and blast. Hem means blood, angio mean vessels, and blast mean to form. So hemangioblasts are named so because they are going to form blood vessels as well as the primitive blood cells. So these are the hemangioblasts, and aggregates of hemangioblasts form a single blood island. But from where these hemangioblasts come? Well, the answer is simple. As you know that this extraembryonic mesoderm, it consists of irregularly shaped, loosely arranged mesenchymal cells. So this is the extraembryonic mesoderm. It consists of these mesenchymal cells, and some of these undifferentiated mesenchymal cells, under the induction of yolk sac endoderm induces these mesenchymal cells. It orders these mesenchymal cells to form the hemangioblast, and these hemangioblasts, they come close together, they clump, they aggregate to form the blood islands. During the third week of development, at about day 17 or 18, this extraembryonic splanchnopleuric mesoderm, that is overlying this yolk sac. So here is the yolk sac, and this extraembryonic splanchnopleuric mesoderm is plastering this yolk sac, and it is almost completely covered with these aggregates, these blood islands. And later on, such blood islands also form within the intraembryonic mesoderm. But the difference between those blood islands and these blood islands that are formed over this extraembryonic mesoderm is that these blood cells of extraembryonic mesoderm, they will form blood vessels as well as the primitive blood cells. But those blood islands that are formed within the intraembryonic mesoderm, they are going to form blood vessels only, not the blood cells. Now here is a single blood island that I have enlarged. Now what happens that some of the cells, some of the central cells of this blood island, they undergo the process of programmed cell death or apoptosis, and so small spaces will form within this blood island. As more and more cells die, these small spaces will become larger and they will coalesce together, and what happens that a central space will be formed, but within that some of the cells they remain alive. Now these alive cells that are within this lumen of this pot-like structure, this vessel-like structure that is now formed, these cells they are the precursor of blood cells. So these are the blood cell precursors, and these peripheral cells that are arranged in the periphery, they are going to form the endothelium of the blood vessel. They are called endothelial precursor cells. They are also called endothelial precursor cells. These endothelial precursor cells, they will differentiate and they will flatten out to form the endothelial cells, and these blood cell precursor cells, they will further differentiate into the blood cells. Now at this point I really want you to have a crystal clear concept of this structure, so let's have a 3D tour. So let's suppose that this is a single blood island, and spaces have coalesced in between this blood island and a lumen has been formed at this stage. Now if we cut a section here, if we cut it from here and we see inside, then what we will see is that here is this blood island, and as you can see that these cells that are in the periphery of this blood island, they are going to form the endothelium, and they are called endothelial precursor cells. And these few cells that are within this within the lumen of this blood island, they are the blood cell precursor cells, and they are going to form the blood cells. So let's move forward. Now of course this is not a single lonely blood island. It has some neighbors, and within its neighborhood there are some other blood islands as well. Now what will happen that these cells, these endothelial precursor cells, they will rearrange themselves in such a way that these blood island, the lumen of these two blood islands, they will coalesce together and a long vascular cord will be formed. Of course, these are not usually two blood islands that coalesce. There are numerous blood islands whose lumen coalesce to form the long vascular cords. Now, what will happen with these vascular cords is that they are the precursor of mature blood vessel. As you know that these blood islands are in the extraembryonic mesoderm, so within the extraembryonic mesoderm, you know that there are the mesenchymal cells, and these mesenchymal cells, they will come and they will surround this vascular cord, and then they will differentiate into the connective tissue of this blood vessel as well as the smooth muscle of this blood vessel. And now a beautiful new blood vessel has been formed, and this process is called vasculogenesis. So vasculogenesis means de novo or new synthesis of blood vessel from the embryonic mesenchyme. Most of the major blood vessels of the embryo are initially laid down by the process of vasculogenesis. Vasculogenesis is an embryological process, and it occurs in the prenatal life, and it rarely occurs postnatally. After the baby is born, then angiogenesis can occur, but vasculogenesis is almost an exclusive embryologic process. So this was the process of vasculogenesis. Now there is another way to form the blood vessels. What happens that certain factors, for example VEGF, vascular endothelial growth factors, they are released. This vascular endothelial growth factor is like a beautiful girl. It stimulates or excites one of the endothelial cell of this blood vessel, and as this endothelial cell become excited, it is converted into what we call tip cell. And this tip cell, it starts to move towards this beautiful girl, and as it does so, these cells, they will proliferate behind this, and they will as a result form new blood vessels. So a whole new blood vessel is formed at an angle by the movement of the tip cell towards the vascular endothelial growth factor. Of course, this is the tip of blood vessel, don't think of something else. This process is called angiogenesis, and specifically the sprouting angiogenesis. The vessel sprouts towards the vascular endothelial growth factor, and this is called the sprouting angiogenesis. Please note that only a single cell will turn into tip cell. If all of these endothelial cells they convert into tip cells, the structure of this blood vessel will fall apart. Fortunately, that does not happen. So, angiogenesis is the formation of blood vessels from existing blood vessels. And this process that we have just talked about is sprouting angiogenesis, and then there is another type of angiogenesis as well that is called non-sprouting angiogenesis that we will just talk about. The angiogenesis is not an exclusive embryological process. It happens in the embryonic life as well as it also occurs during the postnatal life. It plays an important role in physiological and pathological conditions. For example, you develop a wound in your finger or any part of your body, and at that injured part, the blood vessels may get damaged. And to repair those blood vessels, new blood vessels will arise from the existing blood vessels, and that happens due to the process of angiogenesis. Similarly, the tumors get extra nutrition by stimulating the process of angiogenesis within the tumors. So new blood vessels are formed within the tumor by the process of angiogenesis. How can you differentiate the angiogenesis from vasculogenesis? To remember this, you can think that angiogenesis is anglogenesis. What I mean by this is that here you can see that this blood vessel, it is forming at an angle. This new blood vessel, it is forming at an angle. So when a blood vessel forms at an angle, that is angiogenesis. And if a new blood vessel forms from the mesenchymal tissue, that is called vasculogenesis. Simple, isn't it? But remember that this term angiogenesis, it does not mean angle. This term angiogenesis, what it means is that the term angio means, it came from the Greek word called angion, which means small vessel, and genesis means to form, to produce, production or synthesis. So angiogenesis literally means formation of small vessels. So major vessels, they are formed by the process of vasculogenesis, but the smaller vessels, they sprout, they arise from those major vessels by the process of angiogenesis. So as I have told you that this is sprouting angiogenesis, then what about the non-sprouting angiogenesis? Well, it's a simple process. So here is a single blood vessel. Now what happens to this blood vessel is that this blood vessel, it will split into two. So here a sort of pillar forms, and then it is converted into two blood vessels. So this process is called non-sprouting angiogenesis. It is also called splitting angiogenesis, and it is a process in which one vessel literally splits into two blood vessels. I will not discuss the detailed molecular mechanism of this, but the basic idea is that what I think of it is that it is like a mitosis of the blood vessels. As in mitosis, one cell splits into two. In this splitting angiogenesis, the blood vessel literally splits into two. Simple as that. So now let's review what we have studied. So embryonic blood vessel formation consists of two types. The vasculogenesis, that consists of formation of blood vessels from the blood island, and blood islands, they came from the mesenchymal tissue. And then there is angiogenesis, and angiogenesis consists of two types. The sprouting angiogenesis, in which the tip cells are formed and it is stimulated by the VEGF, vascular endothelial growth factor, and the other is the non-supporting or splitting angiogenesis, in which the one blood vessel literally splits into two. Please note that whether it is the process of angiogenesis or vasculogenesis, both of these processes are followed by a process called vascular maturation, and during vascular maturation, the smooth muscle and vascular connective tissue is formed around the newly formed blood vessels. So this was about the vasculogenesis and angiogenesis. Thank you so much for watching this video.